All right, so our next speaker is Pablo. He's coming from the Neurobiology Department and the Segal School of Neuroscience, and he's going to talk about neurovascular coupling in the omic era. Thank you, Pablo.
Thank you, Galit. Thank you guys for staying with us. So I'd like to share most of the work that I've done as a postdoc. I recently, well, it's not so recent already, but I moved to Tel Aviv, and projects in the lab are starting to sort of evolve and mature. So I will be mostly dealing with data from past work that I did. But I will take this opportunity to highlight some key, I believe, issues that are related to the way the Blue Brain project, Idan was here and told us about it, and the Human Brain project will be sort of like approaching data collection. And this is actually talking about big data. And why do we really need this big data? I'll try to show you that with a couple of examples. So even when we talk about the Human Brain project, if you carefully go through the several pages of the first consortium report, you will find out that maybe most of the data collection, to some extent, will be still done in animals, okay, mostly in mice. And if you then carefully read, you will realize that along this year-long roadmap, one of the data sets that we'll actually want to deal with is the neurovascular glial system. Maybe I get a pointer here. Yeah, is this one working? Yeah. So this is key to really understand how the brain works, not only for the purpose of understanding how maybe different imaging modalities work, but this is a key feature of the brain. You try not to use this system, and you probably won't even last for 2 seconds, okay? So a key issue here. Now, I would like also to highlight this word principle. And I think that one of the key issues here is to be able to extract principles about how the brain works from these very large data sets, not just get caught in the details, okay? So if we talk about neurons and the vasculature, I'd like to sort of go back a little bit in history and look at the people that try to answer these questions. And what you can see, this is work coming from 2 British physiologists. This is way back 1850. And they basically were the first to very elegantly show that the 2 systems are actually working together. What they had there was a piston that was, they opened the skull of dogs and they put a little piston that was sensitive to changes in pressure of the PL vessels, the vessels that run on top of the cortex. And then they provide an electrical stimulus to the vagus nerve, and they were able to see this increase in blood flow. This is your very first fMRI machine. So here goes, a few 100 years ago, and we're still a little bit on the dark when it comes to understanding how this actually happens. We nowadays would like to believe that we have way more modern tools that will allow us to actually answer this question. We use 2-photon microscopy. We have awake animals under the microscope. We do chronic imaging where we can follow up the structure, the function of blood vessels, single spines, and there are very specific craniotomy window, but I will not go into the details. So basically what you see here is just one example of how our data looks like. What you see in green is the plasma. Red blood cells flow and they create these dark spots there. And what you see in red are astrocytes that have been labeled with a specific astrocytic marker. Now we're diving into the brain, and you can see the astrocytes and the blood vessels that penetrate and then diverge into the parenchyma of the brain. But we also want to be able to look at what neurons are doing at the same time. So we are now able to pinpoint specific neuronal populations, infect them with a virus that will express a calcium sensor, and by doing this we can actually follow their activity. Of course, you want to be able to do this together. You want to be able to look simultaneously at the activity of cells, neurons, astrocytes, and what the vasculature is actually doing there. So we have the tools now to really tackle this question at the cellular level. How do we translate between neuronal activity to some vascular output? So if another sort of fancy tool that we have today is the ability to use light to actually control the physiology of the system. This is just one example. We can, I'm skipping the details, but we have the ability to control the activity in astrocytes, and you turn another light on, and you can see that the blood flow is changing on that area. So we can use light to both manipulate and observe the system. So I just mentioned that we really want to understand how we translate between these two systems. I'll stay away from the microphone. So into the vascular output, there are many, many different things that go into this transformer function. And, among them, I would like to devote the talk today to vascular architecture. This was it was supposed to take just 1 year of my postdoc work to solve. It took a little bit longer, let me just say so. But anyway, we learned a lot. And let's go for a moment and try to understand what were the physicists' predictions about how a system that needs to deliver anything in nature is actually built. So one prediction, a very dominant view in the field, claims that everything is sort of a branching tree. You go outside, you look at the trees, and you will see this. You go and you look at the leaves, and you will see something very similar. You look at the way the alveolar system is built in our lungs, you will see exactly the same. But a bit of a more realistic scenario, a theoretical scenario, will include actually failures in the system, or will include actually the need to move around resources of limited nature. And all of a sudden, you sort of switch from this binary tree kind of thing into a more loopy network. Okay? So you have two good predictions. Now you need good data to actually test and see which of these two models is actually right, at least for the vasculature in the brain. Now, Idan mentioned before that we have to be able to actually model things. And here's a little bit of a comment that I have on that. Mathematicians will do whatever math needs to be done to show that something is working the way it was presented. These are data-driven experiments. So for experimentalists, it is our duty to provide the most pristine data for this sort of approach. So there was some work that followed that idea of having binary trees, and it's a beautiful description, mathematical description, based on some data. But even beforehand, there were already some descriptions of limited scope in space that show that even through phylogeny, you can have these loop structures in there. So again, we are facing this sort of dichotomy between the two theories. So we just said, enough is enough. Let's just measure everything. Let's just measure the entire PL cortex of both rat and mice and see what is really going on there. So this is exactly what we did as one of the first projects. This is before we had automated tools. We had a lot of undergrads helping us to basically take what you see here. It's a rat brain that was perfused with a specific gel, and it highlights the vasculature. And then you can basically map and translate the anatomy into what we call a graph. This is a network. We have a network of the PL vasculature of both rat and mice, for many rats actually. And what we learn from this is that the network, which you see here highlighted in black, is actually a loopy network. So once you go ahead and you measure everything, once you capture the entire scale of the system, you're able all of a sudden to detect the actual hidden patterns in there. Just as an example, if you were to cut a small window here, you will see a binary tree. So there's a danger there in taking small data. If you do not know, and this is sort of, if you want to remember one thing, this is one of the messages from the talk. If you do not know what is the scale of the feature that you're trying to study, you better take a lot of data. You go beyond what you thought should be your sampling time, your sampling space, whatever. You should go as big as possible given the resources. But nevertheless, you want to understand, I'll skip a little bit of the analysis that you can do there. You want to be able to tie a specific anatomical feature with function. And the way we did that is by following what physicists like to do, where they don't really know how a system works. They try to break it. And the way of breaking this system, or at least perturbating this system, is to actually create occlusions. So you can go and very tightly, you can stop flow in a particular place along these networks. So we did that for this PL vasculature, and something very interesting happens counterintuitively. I mean, if you look at the numbers here and the arrows, they tell you how much blood was flowing before we did the perturbation. And you can actually, once you occlude this place, you can get flow reversals, okay? You would think that blood goes just in one direction. No, it's not true. It can actually go around and come through these loopy networks. And this is not just a peculiarity or some fancy, reorganization of the flow. Whatever we did, we always stayed above this 30% line of flow. meaning that this line is where you start to have actually a pathology. So this anatomical feature has a function attached to it, and this is sort of nice. So what will happen if we just move our zapping laser now away from the PL vessels that you see here, and we now target it to these long penetrating arteries or emerging veins that go and nourish the capillary bed. This is just a tiny difference. Now I'm not targeting the pipes on top of the cortex. I'm targeting the ones that go and dive into the cortex. Shouldn't be a big difference, right? Well, the difference is huge. What you see here on the left is, again, a two-photon image of a flow in this case, it's in red. In green, you see microglia. And you can see that once we create this clot, what you see here is there's no longer any flow there. See what happens underneath. You have all these microglia cells, which are the defense of the brain, basically rushing to create this wall around the piece of that tissue. So we have an infarct there. We have a microstroke. And we just changed a little bit the location of where we do this occlusion. So there seems to be a very particular organization to the vasculature if a small change leads to this extent of damage. Now, can this actually lead to any behavioral difference? I mean, it's just a tiny vessel, right? I mean, what have we done, right? So in order to test this idea, we exploited a system that was, mentioned before by Idan, the fact that we have cortical columns in the cortex. And for those of you not familiar with the whisker system, this is even more advantageous because every single whisker in the snout of the animals, of the rodents, cats and dogs, has a specific representation on the cortex. There's one column that would be the epicenter of the activity if you wiggle that whisker. So you can basically, and of course you have many of them, and you can imagine that you have this big computer at the cortex with plenty of these units one next to the other. So we can train the animals to actually perform a behavioral task just using a single whisker. Without going into details, the animal is in complete darkness. It can use its whisker to measure the distance between two platforms. There's water on the other side. The animal really wants to go and jump to the other side. But if we now go ahead and make this single vessel occlusion, you can knock out the area that was actually responsible for processing that information. And of course, you have a deficit in behavior. So I'm not going into more details. This is NMDA dependent. There's a lot of activity going on there that create this post-ischemic injury. This is for another talk. Interestingly, and this is very recent data from humans, it has now been discovered that the number of these previously unseen events in the cortex are related to conditions of dementia. And this is really, really new data. So we can see that the human is an excellent model for the rats. So, but now, I mean, this strongly established a link between the two systems, I mean, from the pathological point of view. But let's look at a little bit more under normal conditions. What we learn is that there is this notion in the literature of a vascular unit. And that vascular unit is sort of sitting next to a cortical or a neuronal unit. And we really wanted to test this idea because in our eyes, this conclusion or this notion was actually reached using not the most adequate data. Okay? So for the purposes of this, we started by saying, let's map as many cortical columns as possible. So we used a technique which is called intrinsic optical imaging. It was developed here by Amiron Greenwald in the Weizmann Institute many years ago. And you can use light. This is a poor man's fMRI to see where neurons were active based, again, on features from the blood perfusion. And we did that, and you can see that if you select the right portion of the hemodynamic response, you can definitely map neurons using the vasculature. So we then move to ask whether the topology of the system matches at all the place where we actually see neuronal activity. To our surprise, from these data sets, we couldn't find a relationship between these dots which represent penetrating arteries that bring blood into the system and the blue circles that are the places where blood goes out from the system. And we quantified that, and we got to this almost random distribution of these pipes that go into the cortex. So again, we knew the lesson from before. You have to take a lot of data to be conclusive. So we basically mapped the entire somatosensory cortex of these animals to have all the columns and all the penetrating arteries and veins in the surrounding, and we reach exactly the same conclusion. So there's a feature here. If you're looking at where pipes are located in the cortex, you cannot tell where neurons are. Okay, this is important. So again, this is what we basically learned from that, and then we moved to ask whether the organization of the microvasculature might tell us something about the location of neurons. Now, this is where things become tricky, because in order to really answer the question, you want to have in your data sets, you're in front of your eyes in the computer. My kids never understand how I do actually study the brain by sitting in front of the computer. But, I'll have to explain that this is most of our work is done this way. Anyway, so we want to have in the same data set in front of your computer or looking at you, all the neurons, all the vessels, and not just have the vessels, you want to have the network structure of the vasculature there and be able to then compare between the two. So this is what we did. We spent a lot of time collecting a copious amount of data, meaning that we basically took over several cubic millimeters of data per animal with a technique that I'm not going again into the details, but on each data set we have all the neurons in volumes of more than 1 or up to 4 cubic millimeters of somatosensory cortex. We have all the neurons, all the cells, and all the vessels. And again, we don't want to look at the grayscale data. You have to transform it into some meaningful mathematical representation of your data, and we spent several years doing this. But eventually, once you do this process, you end up having a network that represents the vasculature, and you know the location of every single cell in these huge volumes. Now huge in the future would mean an entire mouse brain, which actually exists these days. And once you do that, again, you can reconstruct the entire vasculature over that area, and you know where the cortical columns are. And this is key. We're not guessing their location by any sort of imaging modality. We know exactly where these cortical columns are. Okay, so again, just measuring a large scale, sort of like general features, we couldn't see anything that matches the distribution, the layer distribution of neurons in terms of the organization of the vasculature. Some very basic topological metrics didn't map, again, to the location, the layers of the cortex. And we now measure, again, the loopiness of this network, and we find out that there are a lot of loops, a lot of routes for blood to go left and right. And I'm telling you all this because we wanted to demonstrate that basically the microvasculature looks more or less like a crystal. It is actually not divided, or at least this is what we found. And another way to test that is to basically translate this vascular network into a network of resistors, like the ones that you have in electrical circuits. And there are predictions of how the distance will relate to the resistance along these networks. To make a long story short, a one-dimensional network will scale the resistance with the length linearly. For a 2D network, you go to the logarithm, and for a 3D regular network of resistors, you will end up having a constant value. And this is exactly what we found. We measured tens of thousands of possible pairs of points in these networks, and they asymptotically approach a constant value. So again, pointing to the fact that the vasculature, at least in the somatosensory cortex of mice, is really independent of the location of cortical columns. Well, maybe there's a hidden structure there that we are missing, and we didn't use the right tools. Okay? So we now turn again to what has been mentioned before, network analysis tools, to basically ask the question topologically, meaning in terms of the network structure, can we identify any kind of organization that will point and that would be actually overlap in space with location of cortical columns, or actually we might have a scenario where these topological groups are unrelated to the location of cortical columns. And again, to make quite a long story short, trying many different partitioning algorithms, we eventually found out that the different colors that you see here are the outputs of the algorithms. When you measure the actual overlap in space, if you had a one-to-one map between the vasculature and the cortical columns, all these dots here should be sitting in this null prediction of a one-to-one relationship. And this is not what we see. We actually see that the number of topological groups that are occupying a single cortical column actually increases with the size of the cortical column. Again, you might wonder, maybe this is not the right metric to use. This system, what it does is to distribute flow. It doesn't care about, Facebook friends or any sort of like fancy topological analysis. So, as Idan mentioned before, you have to be able to simulate the reality in your abstracted data. So this is what we did. We simulated blood flow using some basic equations that describe how conductance changes based on distance and diameter. And again, we can then compute this difference in pressure between any 2 points. From that, you know how much flow occurs through every single piece of the network. And then you can run this experiment in the computer where you inject into the vasculature different colors that will basically propagate based on the simulation that you have, and this will determine perfusion domains. And what we observed then, that again, you don't have a one-to-one map between the perfusion domains and the location of cortical columns. So again, we didn't see any match between the two. Now, in order to make sure that you're not saying something that is at least unfeasible, that your model is not actually capturing the essence of the reality, we tested our model predictions with data coming both from our lab and from other labs around that also study these type of systems. And what you see here is just a comparison of the volumes of these perfusion domains to the volumes of the actual micro strokes that we created. So we are sitting on the same ballpark. We are happy. Our model captures the essence of this system. And again, we also capture the dynamics, meaning how long does it take after you make an occlusion to recover your initial flow. So our model captured the essence of the systems. Nevertheless, we were not able to find a link between the vasculature and the underlying neuronal units. So there seems to be a conundrum here. In one hand, I just show you how everything is connected. On the other hand, I just show you that a single pinpoint occlusion can basically create a damage to the cortex. So there seems to be a dissonance here. So again, simple models, if you can put things into a simple model, you basically capture the system. And in this case, imagine that you have, just for the sake of the discussion, you have a one linear array of penetrating arteries surrounded by penetrating veins, and this is corresponding to the ratio that we know that exists in the brain. If you now block one of these penetrating arteries, flow from other sources cannot reach that area just because we have these veins in between that will basically sink out all the blood. So a very simple model that can be extended to 3D. So I will basically finish right now by summarizing what we learned. Basically, you need big data to avoid mistakes on what conclusions you get to. The subsurface microvasculature is really a continuous with no relation to cortical columns, at least in this area that we studied. Again, no topological, no blood flow, support for the notion of a neurovascular column at the system level. Okay? This is key to remember. This is the scale that we look at. It doesn't tell you about lower scale, finer scales. Anyway, so it really points to the fact that you need to understand the microcircuitry of the cortex, how the different cells actually relate and tell the vasculature, we are hungry now, we've been active, we need more blood and more oxygen. So this is where my lab is actually going now, to really dissect this microcircuitry and understand at the cellular level, not at the system level, how we actually communicate between the two systems. And this brings, the approach is again using the available large-scale data. And now I'm, joining forces with people from the Blue Brain Project to actually create predictions. Instead of just going and doing histology without knowledge of what you should expect to see, we put some effort into combining the data that I have obtained during my postdoc with the cellular data that they had at the Blue Brain Project to try to estimate. You don't really see it here. This is a reconstructed somatostatin cell. This is an interneuron, how it should approach the vasculature. We want to have a guide when we go into measuring things to tell us whether what we see is away from, it's actually a random thing or it's actually a directed kind of opposition between the two systems. We're going to attack this while we have some predictions in our minds. So I'd like to finish by thanking all the people that I worked with. This is from David Kleinfeld, neurophysics team in UCSD in California, and most of the work has been done with Phil Tsai, John Kaffel, Per, and Andy and Harvey Carton. Thank you for your attention, and if anything, I'd be open to questions. No questions, it's good. Yeah.
Can you detect, or have you detected any changes in the small vessels following either elastic change or
So you're touching on a very, very hot question in the field. There's actually been a paper published from the Atwood work in Nature this last April, and whether the microvasculature is actually reacting to neuronal activity. This is still an open question. Previous data from Patrick Drew, that was also a postdoc with David Kleinfeld, he showed that the compartment that mostly respond to neuronal activity are the arteries, and this is again opposed to what is called the backpipe model, which claimed that most of the changes actually happened on the venular side. We couldn't find any evidence for that. Actually, we had evidence against that. Whether the microvasculature really changes, you have to be careful, because you need to look at the location of the cells that can actually do this, which are pericytes. So if you measure before, as was done with Patrick's work, anywhere in the microvasculature, you might actually miss the point. Okay, so you have to be able to look at pericytes, and this has been recently done, and the field is not happy with the results, let's say so. Yes.
Talking about scale, do you see all the vessels with your technique? Or do you have a size limitation?
No, we don't have a size limitation. On the well, we might have a limitation in terms of measuring speed, high speeds in the larger PL arteries. We will be limited, and this is because of the scanning speed that you can actually move the beam with current optical setups. But you can probably, if there is an engineered problem, one can go and find a solution. But right now, I would say that in terms of temporal resolution, it's when things go very, very fast that we are maybe missing some of these metrics. But going to the other side, no, we can measure down to these finest capillaries. Okay, thank you.